So we're going to review from the first half of the chapter real quick. So can anyone remind us what an adaptation is? Sam?
It's like a change that an organism develops over time to help it kind of succeed in its environment.
Yep. Just an inherited characteristic from an organism that enhances that organism's survival. And then can someone remind us what natural selection is? Jack?
Over a period of time, only the best fit to reproduce will reproduce, and thus carry on genes that are favorable.
Yeah, so process by which individuals have certain inherited traits that tend to help them survive and reproduce at higher rates than other organisms without those traits.
Alright, so what were the two main topics in Darwin's Origin of Species? John?
Natural selection.
And?
I'm not sure.
Tony, do you know?
No.
Alright,
What
Sam?
was the question?
What were the two main topics that Darwin talked about in his book Origin of Species?
Natural selection and evolution.
Right, so he first started to introduce both of these ideas of evolution and natural selection. What is the smallest unit that can evolve? Sean?
Population.
George.
No.
Oh, God.
Oh, I'm alright. I'll stand. Okay. So we just talked about it, how Darwin brought forth the theory of evolution, and he really just brought the basics into it, and over history, that's been compounded into the tree of life that scientists have been trying to piece together what animals came from where, and we have that. We use the theory of evolution to name new species based on their genus and their species name. So we're going to look at some things that studies that have proved that natural selection occurs, and particularly it comes into play when herbivores' diets are altered. So soapberry bugs are found in two different parts of Florida where the types of fruits that they're available for them to eat are dependent upon their mouthpiece, which acts as a kind of beak to get to the inside of the fruit. So soapberry bugs in varying locations of Florida feed on different types of plants. This is a soapberry bug. And their makeshift beak is most advantageous when its length corresponds with the depth of the seeds within the fruit, and because the fruits are different in the two different locations in Florida, the advantageous length is varied, and natural selection happens. Data implicates that the populations of the soapberry bugs, which if you remember, that's the smallest group of animals that can go through evolution. Their beaks correlate with the fruit that they're exposed to, so when the beak is required to be longer, those populations have an average beak length that is longer, and then the location where the fruits are smaller and it's most helpful for them to have a smaller beak, the length of the beak is, the average is smaller. So we just went through that. So natural selection can be a weighty factor that influences the rate of evolution. So how different it's required for the soapberry to grow is that it affects how quickly the evolution happens and how quickly the soapberry bugs change. Does anybody have any questions?
How can population be like the smallest group to evolve?
Because the population is it's kind of defined by your environment that you're surrounded in. It's a particular environment where there's several factors that change, even in locations as close as just 2 places in Florida. So if there's different fruits in those 2 locations, the population is the group bounded by that area, and that's kind of like the confines of where this evolution is going to take place. Because in that nature, with all those factors, it's going to select traits for the animals to be most suited to that environment.
But like, can't just one organism kind of start to develop?
One organism is the object of evolution. You can trace kind of trace traits through a single organism. Let's say the first animal to walk on 2 legs. Yeah, that was the object of evolution, but it spread to the population, and then the population spread to the world. But a single organism can't evolve because, as Kate talked about with Lamarck, who thought that if a giraffe uses its neck in its life, it's going to pass that trait on so that its offspring will have longer necks. That doesn't happen because genes, as we know, are the same in all cells except for, I think, the immune system. Our DNA is the exact same, and it's only when our gametes go through meiosis that it's changed from crossing over. So it's brought about into new species and, different populations.
I like the explanation. That was as good as I've ever heard with, why an individual cannot evolve. That's the key right there. Only population can.
Now we'll look at an example that involves bacteria with relation to our drug usage, as in medication, and this is artificial selection. So pathogens that resist drugs can evolve into the resistant strains, and the pathogens that are susceptible to the drug's effects die out, leaving the immune type to regenerate the bacterial population. What this is basically saying is that a colony of bacteria, if they're exposed to a drug, will eventually evolve to become resistant to that drug's effects, and they'll find new ways to evade its dying effect. So those ones are the only ones left alive to reproduce, and gradually the colony itself will evolve, kind of like a population, to become entirely resistant to that drug. MRSA is an example in which a particular form, which in this case was a strain USA 300, evolved to become resistant to treatments and more volatile. This is a picture of MRSA USA 300.
Do you know what MRSA stands for?
Something Streptococcus or something.
Yeah, it's methicillin-resistant Staphylococcus aureus. Staphylococcus aureus is a common skin microbe. Staph aureus is on all our skin, okay? It's very common, but this particular strain is virulent. It's more volatile. It's aggressive, and it not only causes infection and eats flesh at a very fast rate, but it's also resistant to all the known drugs, and especially methicillin. And
Yeah, we're going to
so it
talk
makes it
about that.
particularly dangerous.
Okay. So when penicillin was first released for usage, within 2 years of that time, more than 20% of the strains that came into hospitals' care was resistant to the medication, meaning that penicillin had virtually no effect to the drug to help hinder it from procreating. The resistant bacteria had an enzyme that broke down penicillin called penicillinase. And this is a picture of the penicillin transpeptidase and the penicillinase enzyme.
And one other thing I wanted to add in there is that in the microbiology class that I teach at night, we did a antimicrobial susceptibility test and found that penicillin was ineffective against any of the bacteria we tested it on. and it's mainly because penicillin was the first, it's been around so long.
Yeah,
That all the bugs
so-
are just totally used to it.
Yeah, so what Doc is saying is that because it's been in circulation for so long, so many bacteria have been exposed to it and have had the opportunity to evolve. And then those populations are the ones that survive to continue to go through mitosis and repopulate. Researchers in turn manufactured a penicillin which was invulnerable to the enzyme, meaning that that enzyme was defunct when penicillin came into the cell. But within another couple of years, strains have found ways to impede that drug as well. So I think you guys are seeing a pattern where we make a drug and it's effective for some time, and then bacteria eventually evolve, and then we make a new drug to combat that evolution, and then they evolve again. That's kind of how natural selection is working. Methicillin, as Doc mentioned earlier, is a potent antibiotic similar to penicillin, and its circulation began in 1959. It was formidable for 2 years before resistant strains materialized present in the division of
Staphylococcus.
Staphylococcus aureus. This is a picture of Staphylococcus aureus. Methicillin functioned by deconstructing the cell walls of bacteria so that the cell broke down. And it would do this by disabling a particular protein within the bacteria that was used to synthesize the cell wall. So that because that protein was ineffective, the bacteria had no way to build up their cell wall. This is kind of a simple explanation of how this happened. It's really self-explanatory. It just disabled the protein, and we couldn't use it anymore. So, as we've seen, there are resistant strains of Staphylococcus aureus that combated methicillin by utilizing different proteins to combat their cell wall. And the survival and reproduction of this form of Staphylococcus was at a much higher rate than its less competent counterparts, meaning the ones who couldn't synthesize that protein to make up for the methicillin's disabling of their primary protein, died out because they had no way to reproduce and survive. Okay, so the persistence of these strains prompted the spread of MRSA. So because we were using methicillin so much, that evolution happened so often that MRSA became widespread and repopulated at a really high rate and became very dangerous because we had no way to treat it. So MRSA has been an increasingly difficult problem, and it's harder and harder to withstand because some strains have been found to be resistant to more than one form of our drugs. We kind of talked about this in previous chapters, conjugation, how bacteria are able to exchange genes with members of their own and other species. And this has likely led to this cycle of quick adaptation. The MRSA strains known to the present day are likely due to the issuing of antibiotics and the exchange of resistant genes among survivors. A picture similar to this is in our book, but basically, as we see, there's two different bacteria. One is the donor, one is the recipient, and then they converge and they exchange bacterial in DNA, and then they each emerge from each other having a copy of the other DNA. So all it really,
Yeah, keep in mind, it's only the plasmid that's shared.
yeah,
Okay.
the circular DNA, the plasmid. So basically, all it really took was for one cell to evolve this adaptation to any drug, penicillin, methicillin, and then it could exchange that with hundreds of other cells who would have the opportunity to divide because they have been naturally selected to be the dominant strain.